Here we are, we're on our final lecture on organic molecules, and we've saved nucleic acids, things like DNA and RNA, for our final discussion. And remember that nucleic acids, because they're one of the 4 types of organic molecules, do have carbon atoms within them. That's a definition of an organic molecule. And as it turns out, nucleic acids are made up of long strings of nucleotides joined together. So the first question that we're going to address is, what are these nucleotides that are all strung together to make a nucleic acid? And nucleotides are an organic molecule that contain 3 separate discrete parts all joined together in a single molecule. One thing that they have is a sugar group. And in the case of DNA and RNA, it's a 5-carbon sugar, either called deoxyribose if you're talking about DNA, or ribose if you're talking about RNA. And I'll say a little bit more about that in a bit. The other thing that you'll find in a nucleotide besides a sugar group is something called a phosphate group. So this is a phosphorus atom with 4 oxygen molecules attached to it. Okay, so there's the phosphate group. And the final part of a nucleotide, the third part, is a nitrogenous base. And in this picture here that I'm highlighting, it's this hexagonal shaped object in green. Now, the phosphate group and the sugar group in a nucleotide are always the same. But what can be different depending on what type of nucleotide you're looking at is the type of nitrogenous base that's attached to the sugar group. So in the cases that we are going to be talking about DNA and RNA, we have several different nitrogenous bases that could be found in a nucleotide. Cytosine is one, or capital C for short. Thymine is another, or capital T. Guanine, capital G. And adenine, capital A. In RNA, there's another nucleotide I will mention later called uracil. It's very similar to thymine with a minor difference to it. So one thing I'd like you to notice here with these nitrogen-containing bases is they seem to fall into 2 discrete groups. The group above this green line here, you'll notice, are all just a single ring of carbons, one hexagonal-shaped carbon ring. They are called the pyrimidines. Below the green line, you'll notice that these nitrogenous bases, and these include guanine and adenine, you will see there that they have 2 rings joined together side by side. One ring is 6 carbons, the other ring is 5 carbons. These are the purines. Okay, so purines have 2 circles or rings of carbons joined together. Pyrimidines are only a single circle of carbons. So those are the nucleotides and the 3 parts that make them up. Let me give you an example of a nucleotide. Perhaps the best known nucleotide out there next to DNA is something called ATP, which stands for adenosine, because it has an adenine base, nitrogenous base, triphosphate. Okay, adenosine triphosphate. Triphosphate relates to the fact that there are 3 phosphates linked to the sugar group in this molecule. And remember, again, in nucleotides you have 3 parts. You have the phosphate group, the sugar group, and finally a nitrogenous base. And again, as I said, in this case, the nitrogenous base is adenine. And together they make up adenine triphosphate. And adenine triphosphate, or ATP, is a very common energy currency for the cells in our body. It is basically the molecule where energy is stored so that it can be rapidly accessed when needed within the cell. And what makes it so rapidly accessible is the fact that between these phosphate groups, the bonds are very unstable. And I'm using this kind of wavy red line here to indicate that. Anytime you see a wavy line like that, it looks almost like the tilde that appears over an N in the Spanish language. That represents a highly unstable bond that can break apart very readily. And when chemical bonds break apart, energy is released, and we can use that energy inside of a cell to perform work for the cell. So constantly what's happening in your cell is ATP molecules are breaking apart into adenosine diphosphate, meaning 2 phosphates on it now, no longer 3, and it releases an inorganic phosphorus. We use the small I there to indicate it's inorganic because it's no longer attached to a molecule containing carbon. And the other thing that gets released in this process is energy. Now, we consume ATP constantly and very rapidly within our bodies, and if we did not have an ability to recycle ATP, we would run out very quickly and die. So your body is able to, once we've created ADP, it has the ability to go seek out new sugar molecules and, through a process that we will learn about later, extract chemical energy from those sugar molecules and use that energy to reverse this equation to regenerate molecules of ATP. So your body is constantly breaking down ATP to get energy for rapid responses, and then it's constantly digesting sugars and using the energy from the sugar to make more ATP. And like I said, we'll learn more about that when we get to glucose oxidation, which happens in a place inside the cell called the mitochondrion. Let me give you another example of a nucleotide. This is the one that I'm sure everybody has heard of, is DNA, or deoxyribonucleic acid. Deoxyribo refers to the type of sugar that's found in DNA. And if I back up here 2 slides and show you, this picture down here shows you the 2 types of sugar that are found in nucleotides. The one that we find in DNA is this one here, deoxyribose, and you'll notice right here where this hydrogen is, there's no oxygen there, whereas in ribose, the form of the sugar that's found in RNA, there's a hydroxy group, an OH group there. And that's the only difference between deoxyribose sugar and ribose sugar. So back to our discussion of DNA here. Oops. The deoxyribo refers to the type of sugar found in the molecule, and then nucleic acid refers to the fact that we have these nucleotides that contain the phosphate group, the sugar group, and the nitrogenous base. In DNA, there are 4 types of nitrogenous bases you can have: guanine, cytosine, adenine, and thymine. And the way that we join together nucleotides to form a long string called a nucleic acid is via a process called dehydration synthesis. And during that process of dehydration synthesis, I'll write it down here for you, dehydration synthesis, we join together the phosphate and the sugar group. If you look closely at this picture here, you can see along this red backbone, you see the letter S for sugar and then P for phosphate, and they alternate: sugar, phosphate, sugar, phosphate, sugar, phosphate. This creates something called a phosphate-sugar backbone. And then inside of that phosphate-sugar backbone, you can see the nucleotides, the nitrogenous bases of the nucleotides are found in the middle. And I've highlighted those here in yellow. And those nitrogenous bases pair up with other nitrogenous bases in a second string of DNA that's right immediately adjacent to the first string. And they have very peculiar rules for how they join together. The nitrogenous base adenine pairs always with thymine, never with anything else. The nitrogenous base cytosine pairs always with guanine and with nobody else. The easy way to remember this, I figured out when I was in college, the letter A and the letter T both are made all with straight lines, and they always pair together. The letter C and the letter G both have curves in them, curves in the letters, and they pair together. So the straight line letters pair together, the curved letters pair together. Now, this pairing down the center creates sort of, if you look at this almost like a ladder, the sides of the ladder is a sugar phosphate backbone, and the rungs of the ladder going across are these pairings of A's with T's and C's with G's. Now the adenine is joined to the thymine via a special type of bonding called hydrogen bonding that we discussed before. Remember, hydrogen bonds are not true bonds, but they are just associations between partial charges in 2 molecules. So adenine and thymine join together by hydrogen bonds, 2 hydrogen bonds to be precise, and guanine and cytosine join together by 3 hydrogen bonds. And that's what holds the rungs of the ladder together. Now, something else you need to know about the structure of DNA is the presence of these things called 3' and 5' ends. If you look at this picture of DNA here where you can see it spiraled into its double helix nature where it's twisted, which is its natural state, you will notice that down at this end here, one side or one strand of DNA ends with a sugar group. The other side ends with a, actually they've drawn this incorrectly, it should show a phosphate group as being last. Somebody wasn't paying attention in the art department. The end of the strand that ends with a sugar group is the 3' end, ends with sugar. Okay, 3' end always ends with sugar. The end of the strand that ends with the phosphate group last is the 5' end. And in a few minutes, that'll become important as we start talking about how DNA makes copies of itself. But for now, just remember 3' end ends in sugar, 5' end ends in phosphate. And you'll notice that one strand over here and the second strand over here, one ends with a 3', one ends with a 5', which means they are running in opposite directions from each other, or what we call anti-parallel. Okay. Now, we are going to talk about the process of replication in DNA and come back again to this idea of 3' and 5' and running anti-parallel and all of those good things in the second part of this lecture. For now, just keep those terms in mind. I'll also remind you that in our online presence on the internet, in our course room, there's some really, really good animations in there that are going to help you understand the process of replication when we get there. Next up, I want to just briefly talk about who discovered DNA, because it wasn't until the 1950s that we knew what DNA was and what its duty was inside the cell. And there were 2 individuals, 2 scientists by the name of Watson and Crick, who were awarded, along with a third person, the Nobel Peace Prize for their work in describing the actual structure of DNA. Sadly, part of their endeavors to figuring out the structure of DNA involved using this woman, Rosalind Franklin's, research without her permission. They actually found this picture that you see below here, which is basically a radiocrystallography picture of DNA. And based on seeing this picture on her desk, and they didn't have permission to be there to look on her desk, they were able to finally figure out what they couldn't understand about the structure of DNA. That is that it forms this thing called a double helix that twists around each other with the nitrogenous bases joining in the center. And by the time that Watson and Crick and the third gentleman received their Nobel Prize, Rosalind Franklin had died of cancer, and so she was never awarded the Nobel Prize for her contribution to DNA, because they don't give awards to people who, Nobel Peace Prizes to those who are already deceased. So it's unfortunate that she wasn't credited with her contribution to the structure of DNA, but she wasn't. And she was an unusual woman. She was one of not very many women in science at the time. Let me give you just one more example of a nucleic acid. This time I'm going to talk to you about RNA. RNA stands for ribonucleic acid, which means the sugar group that it contains is ribose. It's the one that had that hydroxy group on it. Another difference between RNA and DNA is that where DNA was double-stranded and it formed that helical structure, RNA is single-stranded. So it doesn't do that complementary base pairing where it stays permanently in that state. We will see that it does do some pairing with A's with something called a uracil, not thymine. But we'll get to that later. I'm getting ahead of myself. Now in RNA you have nucleotides for adenine, you have nucleotides for cytosine, for guanine, but not for thymine. Instead of having thymine in RNA, you have a different nitrogenous base called uracil, or capital U. Other than that, the nitrogenous bases are the same. Now, one thing that your book discusses, and you might take a minute to review it, is this idea of something called the RNA world hypothesis. And there are scientists that believe that the first living organisms on the planet may have originally started from molecules which could self-replicate spontaneously. And the only organic molecule we know of that can replicate itself spontaneously is RNA. Therefore, we think that probably the initial molecules that gave rise to all of the life we have on planet Earth were probably originally RNA molecules. All right, let me wrap up part one of this lecture by just doing a quick summary for you about the differences between DNA and RNA. In DNA, the types of bases or nitrogenous bases that are present in the nucleotides include cytosine, guanine, thymine, adenine. Whereas in RNA, we still have cytosine, we still have guanine, we still have adenine, but we have uracil instead of thymine. In DNA, the sugar group in the nucleotide is deoxyribose, that 5-carbon sugar that's missing 1 oxygen. In RNA, the same sugar the basic same structure is used, but the sugar has 1 more oxygen on it, and it is called ribose for the sugar. DNA is always, in its native form, is always a double-strand molecule, and in RNA it's always a single strand. DNA can be as long as a chromosome. It can be millions and millions of nucleotides long, whereas RNA, we will learn later, never gets longer than the length of an individual gene, which could be a few hundred to maybe 1,000 or 2,000 nucleotides, but certainly nothing on the order of 1 million or more nucleotides like we see in a chromosome. DNA is located in the nucleus of the cell and only in the nucleus of the cell, whereas RNA initially is created in the nucleus of the cell, but then it migrates out into something in the cell called the cytoplasm, which is this region of gelatinous-like fluid with lots of molecules floating around in it. That's a good place for us to stop, and we'll pick this up in part 2 of the lecture.